Good morning, everybody. I hate to interrupt conversations, but we do need to get going. And in the interest of keeping maybe to the schedule, which we don't have a great track record of doing that, but we'll we're going to make every effort. So welcome once again to our 6th symposium on Alagille syndrome. And we're so excited that you're all here, and hope that you find this energizing and informative, and that you just have a really good time while you're here. As I said last night, I'm Cindy Hahn, and founded the Alliance 20 years ago. And I have my daughter Elena to thank for introducing me to the world of Alagille syndrome and rare disease when she was born 23 years ago. And this morning, she's actually still up in bed, hitting the alarm clock, I think, half a dozen times this morning. But our journey as an Alagille syndrome family has been challenging, and I'm sure as you all have found, and it continues to have a lot of surprises in it. And the latest is that Elena is being evaluated for liver transplant, something that we never thought was going to happen, but her liver is just deteriorating. So that's our new challenge in life. And you know, as with all the rest of you, I'm sure have experienced that with Alagille syndrome, it's really complex, but whatever's thrown in your way, you kind of find some way of forging through it and coming out on the other side. I wanted to share one of the favorite quotes that I have that seems to describe this moment in life for us and maybe for all of you. And it's anonymous. I don't know who wrote this, but it just says, "You can't stop the waves, but you can learn to surf." And that seems to be what we do a lot of. And, you know, we're surfing the waves and we cross our fingers for the best. And when Elena was first diagnosed with Alagille syndrome, which was when she was about 3 months old, I came across an article by Emily Pearl Kingsley that her son Jason was born with Down syndrome. And I wanted to share this because it was something that I came across and it just really hit home. And I think it really goes also with our theme of navigating a new normal for this conference. And it's called Welcome to Holland. Has anybody heard of this before? Yeah, okay, so I see a few hands. But what she wrote was, "I am often asked to describe the experience of raising a child with a disability, to try to help people who have not shared that unique experience to understand it, to imagine how it would feel. It's like this. When you're going to have a baby, it's like planning a fabulous vacation trip to Italy. You buy a bunch of guidebooks and make your wonderful plans. The Colosseum, the Michelangelo David, the gondolas in Venice. You may learn some handy phrases in Italian. It's all really exciting. After months of eager anticipation, the day finally arrives. You pack your bags and off you go. Several hours later, the plane lands. The stewardess comes on and says, 'Welcome to Holland.' Holland, you say? What do you mean Holland? I signed up for Italy. I'm supposed to be in Italy. All my life, I've dreamed of going to Italy. But there's been a change in the flight plan. They've landed in Holland, and there you must stay. The important thing is that they haven't taken you to a horrible, disgusting, filthy place full of pestilence or famine or disease. It's just a different place. So you must go and buy new guidebooks, and you must learn a whole new language. And you'll meet a whole new group of people you would never have met. It's just a different place. It's slower paced than Italy, less flashy than Italy. But after you've been there for a while and you catch your breath, you look around and you begin to notice that Holland has windmills And Holland has tulips. Holland even has Rembrandts. But everyone you know is busy coming and going from Italy. And they're all bragging about a wonderful time they've had there. And for the rest of your life, you'll say, yes, that's where I was supposed to go. That's what I had planned. And the pain of that will never, ever, ever go away, because the loss of that dream is a very significant loss. But if you spend your life mourning the fact that you didn't get to Italy, you may never be free to enjoy the very special, the very lovely things about Holland. So, welcome to Holland, everyone. And more specifically, welcome to your new normal. That's Alagille syndrome and all its complexities. We're here to help you navigate your new normal, and we're starting right now today with a lot of information. We know you might feel overwhelmed and exhausted by this event, but we hope that you'll also be energized and encouraged by the people that you meet. Alagille syndrome is challenging, but with the support that we and everyone here this weekend are able to provide, our goal is that you're equipped to forge boldly forward into your new normal with strength and optimism and hope for the future. So, to start off this morning, we have 3 researchers here to talk about different research studies that are going on with Alagille syndrome and how you can participate in those. And first up is Kira Kennedy, who is with Lumina Pharmaceuticals, recently acquired by Shire. And she is going to talk about some new treatments that they are developing for Alagille syndrome.
Okay. Well, thank you very much this morning. I'm delighted to be here this morning. My name is Kira Kennedy. Up until about 2 weeks ago, I was the Chief Operating Officer of Lumina Pharmaceuticals, a small San Diego-based company. And now I'm the Head of Cholestatic Liver Disease Research, which sounds a very grand title, of Shire Pharmaceuticals. So I am here today to tell you about what our companies have been up to in Alagille syndrome over the last couple of years and what we look forward to doing in the coming years. Cholestasis really centers around, for us, centers around bile acids. So impaired bile flow. I'm sure many of you who have your children have liver disease, you hear about paucity of bile ducts, so fewer bile ducts, smaller bile ducts, which leads to a reduction of bile flow into the intestine and a buildup of bile in the liver and in the circulation. And that's really what we are centered on in terms of a treatment for this disease of cholestatic liver disease. What we are focused on in the intrahepatic liver diseases, including Alagille syndrome. We do have active programs in other liver diseases, PFIC, pediatric disease, and other adult programs as well, but all of what I'm going to tell you today is really focused on the Alagille syndrome work. So clinical research. Like many things in life, and I would say almost everything in life today, it's really a collaborative process, a collaborative effort. In general, in rare disease, it often can start at a small company, a company that's intensely focused on one mechanism or one drug or one disease. And that's really what Lumina was. Focused on the liver. Everything was focused on bile acids and how we can merge those two things into potential treatments for patients. But we're not in this alone. We work with the Children's Network. I'm sure many of you know our for your management of your care that are in the network, CHOP, Pittsburgh, Cincinnati, all the big centers that treat a lot of patients with Alagille syndrome. And along with the Children's Network comes the NIDDK funding the network, making sure that the infrastructure is there to conduct research is a critical part of syndrome. Many ways in which we wouldn't be where we are today without the collaboration and help of the Alagille Syndrome Alliance. An organized patient advocacy effort goes a long, long way in any type of research, but particularly in rare pediatric research, to really coalesce the community around the disease and around the research that's ongoing, as clearly by everybody sitting here today. But without the families, there's nothing to study. There's no families to interact with. So almost at the epicenter of any of this research are you, patients, the families, the Alagille syndrome community. Many of you may be you're aware of or unaware of, you probably have already contributed to the research that is ongoing in Lumina, and I'm going to tell you a little bit more about that today. Lumina is a small company, but we were fortunate to have interacted with a very large and forward-looking visionary company like Shire. Shire is a company that is dedicated to advancing rare disease research. They have the infrastructure to conduct global clinical trials and get this product, should it prove beneficial, out to patients all over the world, which obviously is critically important. Lumina was a 17-person company at the end. We had 9 clinical trials going on in 5 different indications with 2 products. We were exceedingly busy. I don't think our feet really touched the ground very often. But now we're a company, and that just gives us a lot. Something that struck me from the beginning of our interactions with Shire is that they coalesce around as a company, as the people we help. So we can all work hard, but at the end of the day, we go home. And it's the patients, the families that live Alagille syndrome 24 hours a day, ride the waves and learn how to surf, that are the bravest of all. And I've been so inspired working in this ever had because of the people who are in it with us. So when a company like Lumina is starting off research in that, it's not experts. We talk hours and hours talking about this disease, about how it's treated, about how it manifests, about what the treatment options are, where the deficits are in terms of treatment options, and the handheld? Sure. Let me just take this. That's okay. Apparently I wasn't speaking loud enough for everybody to hear, so hopefully this will be better. So as I said, we've spent a long time talking with experts in the US, in the UK, all over the world. And Alagille syndrome, both from a physician and really from a patient perspective, doesn't really differ about much. We have found there's nothing special about US patients versus UK patients. It's really a global disease, and that's not always the case. It's helpful in terms of clinical research when there's more uniformity, and that's certainly what we have found in our research. Very importantly, we spend a lot of time focusing on the patient perspective. Nobody knows this disease as well as you do from your viewpoint and your daily interaction with this disease. So again, many hours of discussion with Cindy Hahn and the Alagille Syndrome Alliance team in terms of understanding the disease, understanding the patient voice, what they're hearing about what families need, what patients need, where the gaps are. We have leveraged data from surveys that the Alagille Syndrome Alliance has put up and delivered to the community. We have leveraged data from the LOGIC study, the genetic study that's ongoing through the Children's Network. And that's why I say many, if not all of you, have participated in this research in some form or other. We have conducted our own interviews with families. We have conducted probably interviews with about 70 to 80 families across the world, and that has led to the largest database of detailed information around Alagille syndrome, in particular the symptoms associated with the disease, itching, and the impacts of those symptoms on your daily life. We also spend a lot of time talking to FDA. We can go off and do a lot of research, but if FDA is not aligned with that research, then it all goes for nothing and we don't get the drug approved and make it widely available to the patients who need it. So we spend time talking to them about what we're doing, how it's going, what we plan to do, and I would say it's been a very collaborative, productive relationship in this instance, and I think that's to a large degree helped by the fact that Dr. Andrew Mulberg is the deputy director of the division that we work with, and he is a pediatric hepatologist who did his residency at CHOP, has treated Alagille syndrome patients, has treated Cindy's daughter. So he has an intimate knowledge of this disease, which isn't always the case. I mean, FDA they're human beings just like the rest of us. They're doing their job and they're dealing with onslaughts of information. In this case, we have an asset and a resource there for us who understands this disease in a way that is unusual. So with FDA, normally you have to educate the FDA specifically about this disease. We still have to go through that process. We still have to support that process. While Andrew may be a supporter of ours, we still have to check all the boxes that FDA requires. And we talk about study endpoints and what we will measure and how we will measure it, and I'll talk about that in more detail. So one of the things that when we started doing research into Alagille syndrome and the particular mechanism that we were evaluating, we looked to the literature. We looked to how bile acids have been regulated in the field of pediatric cholestasis and what that tells us. I'm sure some of you, and we're going to hear more about this from Dr. Piccoli, some of you have heard of or maybe gone through the experience of a partial external biliary diversion surgery, really to drain bile acids directly from the gallbladder into a bag. So you're sucking bile acids out of the system. And what that results in is reduction in bile acids, which is what you would expect. And that is associated with an improvement or reduction in bilirubin, so the liver is getting healthier, and a reduction in the symptoms of itching. So this, to us, is really encouraging data. The data is very comparable, generally, from Alagille syndrome and PFIC, one of the other pediatric diseases I mentioned earlier. So this is data that gives us incentive to move forward with our mechanism, which is a drug that inhibits the bile acid recycling through the enterohepatic system, which we'll hear more about. So this is really the impetus and the data that says, yes, we think that there's a good, solid scientific rationale to move forward with clinical research, preclinical research, clinical research with this drug. Another source of data for us was the biliary diversion survey that the Alagille Syndrome Alliance posted on their website that many families responded to, 37 respondents, in fact. And it may seem like you fill out a survey here, you participate in a registry there, maybe you're part of Logic, maybe you're not. I just want to reiterate today, if I make no other message clear, I want to reiterate that all of that information that you have provided has led to the protocols that we have developed that we are executing in the clinic, the endpoints that we are looking at, the discussions that we have had with the FDA and other regulatory bodies, including the MHRA in the UK. So whilst it may seem like you're putting information into a black hole sometimes, that information is working for you. So you may or may not have seen the analysis of that data, and this is purely our analysis. But what we did was we looked at every single respondent, where they were before their surgery in terms of itching on a score of 0 to 4, and where they were after the surgery. And generally what you see is itching gets better, whether it's the caregiver who's responding or the patient who's responding. So again, this shows us if you can remove bile acids out of the system, symptom relief occurs with this surgery. An important thing when we were thinking about a clinical trial, how long should a clinical trial be? How long is a piece of string? You want to have a trial that's long enough to give you enough time to see a change, but you don't want to have a trial that's longer than it needs to be because you have patients who are on placebo in that trial, and you want to make sure that you're minimizing the amount of time that any patient is exposed to a placebo drug before, in our situation at least, they can get access to the drug. So what we see here is within a month, half of the patients who respond within 1 month. The bulk of the rest respond within 3 months of their surgery. So this is critical information. Our randomized placebo-controlled trials are 13 weeks long because of this information. So there's a real direct correlation here with how we're doing our research. Another aspect is xanthomas. So obviously, some Alagille syndrome patients have xanthomas, some don't. The patients who did have xanthomas, 70% of those patients saw a reduction in xanthomas after the surgery. But the time frame's a little bit different. So it takes a little bit longer for the xanthomas to resolve after surgery. 27% saw a change within 1 month, some more by 3 months, additional ones by 4 to 6 months, and sometimes longer. So we are evaluating xanthomas in our long-term extension study. It's a 48-week study, so that gives us the time to see whether we're going to see a change in that particular presentation of the disease. I'm not going to go through this slide. I will make the slides available. I'm sorry I wasn't organized enough to get them out to you in advance, but we will make them available. These are really just a summary of the observations. Itching, following the surgery, itching goes away, and it's generally gone within 3 months. Xanthomas decrease. It may take a little bit longer. There's a strong link between reduction in itch and improvement in quality of life. Again, not exactly rocket science, but here's some data to support it. And this is data that we used in discussions with FDA. And partial biliary diversion surgery, it brings with it its own complications and risks. So if there was a once a day in the morning oral solution that achieved the same results as surgery, then that will be an ultimate benefit to the patient population. And that's exactly what we are trying to determine if that's what our drug will do. So we have, I mentioned we've done research with Alagille syndrome families. We've sort of been like a traveling community, tromping around the US over the last 2 years. Started off with the Gayman's Picnic in Pennsylvania. We were at the virtual walk in Cape Cod, the family days in Orlando and North Carolina and Los Angeles. Interesting story, and everybody, the world is so small today. I remember seeing the movie 6 Degrees of Separation and thinking, no way, there's no way there's only 6 degrees of separation. I actually think now it's probably less. At the November 2012 Alagille Syndrome Alliance Family Day, I was in the room and I heard a familiar accent. So my accent may not be a giveaway, but I was born and raised in Ireland, in Cork. And there's a family from LA. Do you mind me identifying? Michael O'Shea and I grew up a few blocks apart. My brother went to school with Michael, and we met at an Alagille family day in Los Angeles. His daughter's name is Kira, who has Alagille syndrome, and she is, in fact, the poster child for our itch study. So even though we may not start out being connected or understanding each other in terms of our disease, your disease versus our research, ultimately, we're all connected, and now I very much feel like I'm part of the Alagille syndrome community, if you'll have me. So this was the first wave of research that we did. And the results of this research were presented to FDA in a 2,000-page packet, I kid you not, it was this big, of research to the agency to understand what are the symptoms associated with Alagille syndrome from a patient perspective, from a family perspective, because FDA really cares about that, and we'll talk about that a bit more in detail when David is giving his presentation. What are the impacts of those symptoms? How do you measure those symptoms, and how do you put that all together in an instrument that you can use in a clinical trial that will assess whether a drug is having its effect? So we made some changes to the instrument based on discussions with FDA, so that meant we had to go back to the drawing board, and these are all the places we went to. Cities all over the country, states all over the country, to meet with families of Alagille syndrome patients and PFIC patients to understand, to make sure that the changes to the instrument didn't change the way it was perceived and understood by the families. And we didn't stop there. We've also taken it international. We have met with families, Alagille syndrome families in Spain, France, Germany, Poland, and US Spanish-speaking families in the US, because not only do we want to do our research in English, we want to do it in a lot of other languages too. Now, obviously, there's lots more languages to go, but we're a small company, or at least we were a small company, so we were taking bite-sized chunks of this puzzle. But as you can see, this is a global effort, and this has led to a huge database of research about Alagille syndrome, and we're just now starting to publish and present that data. So not only does it help decide whether the drug that we're evaluating in clinical research has an effect, it provides a body of research from a patient perspective that will be added to the scientific literature for other researchers and regulatory bodies to draw upon. So the first set of data, the really descriptive set of data, we've submitted for presentation at the American Association for the Study of Liver Disease conference that happens later this year in November. So I've mentioned our drug. I've mentioned probably that it's called LUM-001. So what is it? What does it do? So bile acids are secreted from the liver into the GI tract, and then they are essentially sucked back through the enterohepatic system back to the liver via the apical sodium bile acid transporter. So it's a transporter in the wall of the GI that is essential for scavenging bile acids back to the liver. It's very simple. Our drug blocks that. So the bile acids don't go back to the liver. They go into the colon and they get excreted in the feces. So many of our researchers or physicians describe it as a chemical diversion. So you're shunting bile acids out of the body just through a different mechanism, not through a bag or a stoma on the abdomen, but through the stool, through the natural process of the stool. This drug has been shown to lower serum bile acids, both in animal models and in patients, in healthy volunteers, in patients with high cholesterol, and we are evaluating it in patients with cholestatic liver disease right now. I'll show you some data in the next couple of slides about an animal research model that we have completed to show that there's actually, it looks very promising, at least in the animal model. The animal models are not perfect, but they at least give us some more information. One important piece, particularly for patients with liver disease, this drug was designed to not be absorbed into the blood system. Most side effects that happen with other drugs is because the drug gets somewhere you don't want it to go. It gets metabolized in the liver. It has an off-target effect. The target is our target's in the gut. The drug stays in the gut by design, and then it's excreted. So it's not going anywhere it has no business to be. And that actually was designed that way to have a more beneficial safety profile. In the subjects, this drug has already been in 12 clinical trials, apart from our research, 12 clinical trials, over 1,400 patients and subjects exposed. And it has been demonstrated in those patient populations to have a good safety profile. What does that mean, a good safety profile? It means that there are no serious adverse events associated with this drug, or very, very few serious adverse events associated with this drug. The primary adverse event is actually mechanism related. By increasing the amount of bile in the colon, you can have a gastrointestinal effect. You may have some nausea and cramping and diarrhea because your bile is a detergent. If you put a detergent into a system, it's not going to be neutral. But we see this as almost evidence that the drug is working in one way. It's that those effects are transient and they're almost always mild to moderate in nature. So in terms of the trade-off between serious liver disease and a short-term AE profile that affects patients for a limited period of time, we think that that's a reasonable risk-benefit. But ultimately, that decision will be made by the patients. And 001, or LUM-001, is currently in clinical trials for Alagille syndrome, progressive familial intrahepatic cholestasis, primary biliary cirrhosis, and primary sclerosing cholangitis. All cholestatic liver diseases, all with the common characteristic of elevated bile acids. The model that we have demonstrated some effect in is, there's no great model for Alagille syndrome specifically, but what we have done is we essentially tied a cord around the common bile duct to reduce the amount of bile that gets out of the liver. So by, in essence, sort of trying to replicate that paucity of bile ducts that's present in Alagille syndrome. What happens when you do that is, when you look at the stool concentrations of bile acids, you see, this is the normal animal, you see a reduction. So there's less bile getting in the colon, therefore there's less bile in the feces. When you treat with this drug, you see huge increases in fecal bile acid excretion. So you're blocking the ASBT. The bile acids are not recycling back through the enterohepatic system, and they're being excreted in the stool. What does that mean when it comes to biochemistry? I'm sure these are all parameters and analytes that you hear about all the time. if you don't treat the drug, if you treat the animals with a placebo, you see huge increases in serum bile acids. So the bile acids have to go somewhere. They can't get into the colon or the gut, so they go into the serum and they're elevated in the liver. Looking just at the orange bars, what that leads to is increases in GGT, in bilirubin, alkaline phosphatase, AST, and ALT. And when you treat with this drug, you essentially, in this model now, you normalize the biochemistry across the board, particularly at the high dose. So this is really exciting research. Similar research to this was presented at AASLD last year, and this particular research will hopefully be presented there this year. What does that mean to the liver when you get to the tissue? So this is the treated animals, and I'm sorry to get too technical on this, and I'm happy to discuss this in more detail with anybody afterwards. This is the liver section from the treated animals. So you essentially have a normal liver, what looks like a normal liver here. You see the intact bile ducts. You see very few inflammatory cells. In the animals that were not treated, all these dark cells that are not present over here, they're being stained up because they're inflammatory cells. So you've got a lot of disease activity going on here in the untreated animals. So again, this is all very encouraging that this mechanism may actually do something in the clinic. So what do we think that this mechanism can offer in terms of potential? Now this is all potential. We don't have data yet in Alagille syndrome that we can read out on. We have ongoing studies, and I'll tell you about those. But we think that there is the potential for this drug to improve liver function, to slow disease progression, to reduce itching, and ultimately to improve quality and hopefully quantity of life. So what are we measuring in our studies? Serum bile acids, itching, and we look at itching in a variety of different ways. We look at itching from the patient report perspective, from the caregiver perspective, and from the clinician perspective. So we're coming at this from every angle of everybody who's involved in the care of your children. We're looking at how is the liver functioning? What are the markers of liver injury and liver health? Bilirubin, ALP, AST, GGT. We're also looking at fat-soluble vitamins, xanthomas, and quality of life, and a variety of other things that we look at for safety. We want to make sure that we're not improving one thing, but having a negative effect somewhere else in the body. So we look at a lot of safety parameters. So what are our studies? We have 4 studies in Alagille syndrome right now, and I'm just going to pop them all up here. And these are all Alagille syndrome patients. Some of them you may recognize, some of them you may not. Erin lives in the UK, Eunice here in the US, Kira in the US, and Sophie lives in Australia. So Imago is the first study that we opened. This is the study that we're conducting in the United Kingdom. We're getting very close to completing enrollment in this study, which means we should have data from a placebo-controlled, blinded study, real data that we can interpret in a meaningful, objective manner by the end of this year. When patients finish Imago, they can participate in Imagine, which is the 48-week extension study where all patients get drug. And you have the opportunity to optimize your dose based on your symptoms. So far, touch wood, all patients that have finished Imago have rolled into Imagine. So we have a really good track record here in terms of patients at least wanting to have their life disrupted enough by clinical research to keep on going. And that's no mean feat. I mean, these extra visits and traveling to sites and dosing every day and doing electronic diaries. I mean, this is not an insignificant impact on life. Itch is a study that's very similar in design to Imago that we will be conducting with the Children's Network in the US. We are getting close. It's been taking a little longer than we would have liked, but we are getting close to being able to enroll patients in itch. And again, patients that complete itch can enroll into Imagine. So even if you are randomized to drug in the first study for 13 weeks, you will have the opportunity to go into the Imagine study and studies thereafter. If we're continuing to develop treatments for Alagille syndrome with this product, there will be no end date to your treatment until this drug is available commercially. An Iconic is a study that we are executing in Europe, in Spain, France, Poland, Germany, also in Canada and Australia, to serve those patient populations and make sure that what I mentioned earlier in terms of all Alagille syndrome patients, do they respond the same way, that's true for not just the US and the UK, but for other countries as well. So just wanted to sum up in terms of that our role in research is clear. We come up with the drugs, we make the defense, we go to the FDA, we get approval to do studies, we conduct the studies. We can do all of that, but without you, that's all for nothing. So your role in research, and one point that, again, I wanted to make clear that resonates is the information you share really matters. So whether it's surveys or interviews with us or others or the registries, it really advances the understanding of Alagille syndrome. It has shaped and molded how we have designed our clinical trials. It helps us develop the right instruments to measure the symptoms that impact your life in the most meaningful ways. And it provides a platform for discussions with FDA, which ultimately, and EMA and every other regulatory agency in the world. And participation in clinical trials. My goal is not to get every family to sign up for a clinical trial. My goal is that every family is aware that there is an option in terms of clinical research. So the awareness piece comes from us. The be informed piece is up to you. If you're interested, we will give you all the information you need to make that decision for you, your family, and your child. And consult with your physician. So the doctors that are involved in our studies are very well informed about this drug, about the protocols, about what it will mean for participating, and whether it's a good fit for your family and your child. So I would say your first protocol, I mean, we are certainly here to give you information, but your first protocol should be your physician and talk to them about the research. If they're not participating in the research, we will get you into the study if that's something you want to do. We will support patients who are willing to travel to centers who are in ITCH, for example, in the US, or anywhere else to the degree we can, and just talk to us. Don't be afraid to come with a proposal to us in terms of your participation. And I will finish with a thank you very much. Very happy to be here.
I think we might just forge ahead here and have Alexandra Falsey from CHOP come up and talk about the study that they have ongoing. And then we're going to go to Pedro Sanchez about the hearing study that they are doing. And I did want to mention that out in the hallway, we have sign-up sheets for participating in talking with Lumina, also doing hearing tests with Pedro and Gage Crump. And I believe that CHOP is also doing blood draws. Yes, okay, for their genetic database. So, without further ado, I'm going to bring up Alexandra, and we may have to do a switch out on the, okay.
Thank you so much. Yep. Great, so while Chris is getting that loaded up, we'll do a quick introduction. My name is Alex Falsey, and I am the Clinical Research Coordinator for a genetic study at the Children's Hospital of Philadelphia under the leadership of Dr. Nancy Spinner.
And my name is Chris Kurchowski, and I am the Senior Research Technician in Dr. Spinner's lab.
So as Dr. Piccoli introduced yesterday, Dr. Spinner has been working on Alagille syndrome for a number of years now, and actually her lab was the one to find both Jagged 1 and Notch 2 and their role in the Alagille syndrome. But one of the questions that we have not yet answered and we're still trying to figure out is, how is it possible that individuals within families can have the same Jagged 1 mutation but can have very different cases of Alagille syndrome? So in genetics, we call this variable expressivity. So the question is this just a random variation? Is this just something that happens? Or are there other genes that could potentially be involved in the pathway and in terms of the clinical outcome? So to try and answer that question, we have looked at our wide repository of samples from people with Alagille syndrome, and we're curious to compare individuals with Jagged 1 mutations and look across the rest of their genetic code, the rest of their genome, to see if there are any other regions that could contribute to their liver disease severity.
Okay, so to do this, we did something called a genome-wide association study. And that basically means that we took two groups of people. So in this case, we took individuals with mild liver disease and individuals with severe liver disease. And across both groups, they all had JAG1 mutations. And so what a genome-wide association study does is we want to see what's the difference between the two groups. So we know they all share a JAG1 mutation, but are there other regions of the genome that they have in common or different that the two groups share together? So when we did that, we ended up finding a region of the genome that was close to a gene called thrombosponin 2. And this gene has known interactions with the Notch signaling pathway. So then we wanted to see what basically, so what is thrombosponin? So it's a gene that is affected in cell adhesion and migration. And why we originally wanted to zero in on this region is because we know that it has known interactions with the Notch signaling pathway. And so we kind of wanted to look more into how this gene might interact with a JAG1 mutation or interact to create either a mild or severe liver phenotype. Okay, so our first question that we asked is thrombosponin in the right place? So if it's expressed somewhere outside of the liver, that's not as interesting. So we wanted to see if it is expressed in the liver, and specifically if it's expressed in portal tracts or mouse bile ducts. So we started in the mouse, and we wanted to do a staining. So I know it's a little bit hard to see. But basically, we were able to see that thrombosponin is present in mouse bile ducts. So from there, we wanted to move on and see more specifically if it's a different expression in the mouse portal tracts versus the rest of the liver. So we did a really cool experiment where we took a mouse cell, so a mouse liver cell, and we laser captured out just that portal tract. Okay, so we had just the portal tract, and then we had the rest of the liver cell, and we wanted to see, is there an increased expression in thrombosponin in that portal tract versus the rest of the liver? So when we did that, we basically saw that there is about an 8-fold increase in expression in the mouse portal tract versus just the rest of the liver cell. So we know that thrombosponin as a gene is in the right place in high levels.
So just to take a step back and provide a little extra context, when you do have all of your genes, you have about 20,000 genes in your genome, so all of your genetic code. And not every gene is expressed in every place in the body. So as Chris was saying, it's particularly interesting that this thrombosponin 2 gene is expressed around the portal tracts in the liver. It gives us an indication that this could play an important role. So we've done a lot of studies in the mouse samples, and so this is where you step in and hopefully can help us out. So we really are interested in seeing how does thrombosponin 2 express in human samples. So unfortunately, thrombosponin 2 is not expressed in blood. It just isn't. Unfortunately, the only place it is expressed is in fibroblasts. So that would require us to obtain skin samples. And actually, Dr. Lin is here, and he can help us. So we've got a sign-up sheet outside. It's really not a painful process, and this could help us to figure out what role this plays in human disease. So if you have any questions, we are available. I promise we don't bite, and we're pretty good at answering any questions you might have. So please don't hesitate to ask us.
So just going back on that, so I tried really, really hard to get this to work in the blood. It's just not expressed in the blood, and so I can study a mouse sample as much as I want, but it's much, much, much more interesting to look in humans. And so getting that skin fibroblast sample is really, really important because it allows us to see specifically the people who have JAG1 mutations, how is thrombosponin look? How is it expressed differently in different people? So it's really, really, really important that we can get fibroblasts specifically. So fibroblasts are just skin samples. So it's really, really easy, and it would really help the research a lot. So just on a completely different path, so one other pilot study that we've just launched in the lab is to do whole exome sequencing of people who are JAG1 and Notch2 mutation negative. So whole exome sequencing is a relatively new genetic procedure that allows us to look across someone's entire genome at every single gene. So if you think about JAG1 and Notch2, so those are two genes that everyone has, but people also have about 20,000 other genes, a little more. And so whole exome sequencing allows us to look at every single one of those genes. And so since 4% of individuals who are clinically diagnosed don't have JAG1 or Notch2 mutations, doing whole exome sequencing allows us to see every other gene to see if they possibly have mutations in anything else. So again, we've launched a pilot study. Right now, we've done about 3 families, and we are in the initial stages of validation to look at some very interesting variants that we found.
So I think that sums it up. I know this has been very science heavy, but please don't hesitate to come ask us any questions. We'd love to break it down for you and explain a little bit further. But we are here on behalf of Nancy Spinner, who unfortunately couldn't be here this weekend, but she was very sad to miss it. So our team really is very heavily populated with some amazing people. So Nancy Spinner, Kathy Looms, who also couldn't be here, David, who's sitting back there, Marcella Devoto, and we definitely couldn't do this without support from the NIDDK. So thank you so much, and please don't hesitate to reach out.
And again, I'm totally willing to, if anyone wants to see, I'll do one on my own, or Henry will do one on my own arm. It doesn't hurt at all. It's completely numbed, and it's just a tiny little, it's less than a fraction of your fingernail. It's just a small little punch right here. It just takes a tiny skin sample. So more detailed information, I think Dr. Lin would be able to explain, if you'd like.
Good morning. Pleasure to meet everyone. Sorry I couldn't make it last night. The skin biopsy stuff sounds pretty intimidating and ominous, and I agree. I actually thought the same thing the first time I did one. But we've been doing this for quite some time now, and actually what it entails is, as Chris was describing, it's a small punch biopsy. Essentially, it's less than half a centimeter across in diameter, is about how big of a biopsy or hole that we're making in the skin. It's not very deep at all. It's probably about the same amount deep. So if you think about it, it's like 1/4 of an inch by 1/4 of an inch tops. It's a small little circle that actually, you know, what happens is we numb up your arm. We take it kind of in the fatty part of the arm right there, or right here. I don't have any fat. Hey.
That's okay. We're friends.
So you take it in the fatty part of the arm here, you numb it up, and then you take the skin biopsy. It's actually such a small biopsy that you don't even need a stitch to sew it up, and actually you put a Band-Aid over it with Neosporin, and it should heal up. But I think that we have a few comments, questions? Yes, at the picnic. Yeah. There you go. Oh, so we just had a few of our prior skin biopsy volunteers who are volunteering to show the scars if there's any questions, actually. Yes. Yes. Needle for the numbing.
And again, the more samples that we get, the more interesting conclusions that we can draw from our study. So really, really like as many people who are willing to participate, to hopefully.
All right, so our third talk right here is Pedro Sanchez from Los Angeles and his trusty companions, B. Smith and Gage Crump.
Thank you, guys. We'll take a couple minutes just to describe what we're doing here. I also have ties from CHOP. I trained with Nancy Spinner and Ian Krantz and David, and I think my heart is always tied to Alagille syndrome, so I keep coming back. But now I'm at Children's Hospital Los Angeles, and I work with Gage and B. And this study was sort of inspired by Gage because he really studies development and basic science in biology of facial development. And he has some great animal models that we could test a lot of these hypotheses and actually discover new things in these models and see how we could help patients in the long term. So he has some models in zebrafish and mouse models, and he's going to give a talk a little bit more detail later on in the second session in the afternoon. But it talks about evolution and fish biology and how the jaw in the fish actually evolves into the middle ear bones in your ear. And so I'll show a couple quick pictures, but the question is, if you have a genetic change in the JAG1 gene, and there's a potential middle ear defect in the bones, can there be hearing loss? And in fact, we know that everybody can hear pretty well, almost similar to everybody could see pretty well. But a lot of us have glasses, and if you take off your glasses, you probably can't see the board. And so, I'm not technically blind, but I won't be able to do as well in school if I can't see the screen very well. And so the question is, if there's some mild hearing loss, maybe there's a way to help those kids a little bit more and amplify them. And we did come to a meeting last time, and we tested several families, and I'll show some of those results. But this is some of the pictures that Gage will describe later. And the question is, we tested it in zebrafish and in mice, but similar to the CHOP studies, is really the most important question is, how does it influence humans? And so we come here asking, is hearing loss, do kids have hearing loss or adults have hearing loss? And how common is it? Should there be additional recommendations? And should it be monitored or treated? And then once we do a hearing test, we'd probably recommend somehow imaging the bones in your middle ear with a CAT scan. And a couple people have followed up with that since the last study that we did 3 years ago. So what we're going to do here is Bee's here. She's an audiologist, and she'll be conducting a hearing test. And she could describe that in a second. We'll also get a saliva sample just to get some DNA, just to confirm the genetic change in Alagille syndrome. And also, if you do have hearing loss, test the hearing loss genes just to make sure there's no confounding factors, or maybe there's a modifier of that effect. And we'd also recommend a CAT scan if you do have some hearing loss. And the nicest thing is that Gage and I have applied for funding, and we do have some funds from the March of Dimes to complete some of the animal studies and to actually bring some people to Los Angeles if you don't have a local person that's advocating for you to get the appropriate care for your hearing and the scans. So if you're local or you're just passing through Los Angeles, we'd love to see you. And these are some of the tests we did last time, and B can just describe what we could see here. This is actually not caused by Alagille syndrome, but this is from noise-induced hearing loss from someone who actually was listening to loud music during their teens. So, and so it's a complicated little chart that we'll be drawing, but we're going to not only do the test, but explain the results to you sort of in a confidential manner. Last time we tested 19 subjects. We have 22 people tested. Half were normal, half were abnormal. And so, it's a subtle change, but we're really trying to figure out the influences of Alagille syndrome and hearing loss. We're looking for conductive hearing loss. And so when you hear conductive hearing loss, people say, oh, that's because of fluid, and maybe that'll go away. And so we do 2 parts to the test. There's a tympanic membrane test where we look for the movement of the eardrum. And if it's conductive and it's not moving, maybe that's true, it's fluid. We'd want to repeat that. And if there's no fluid there, then it actually might be a middle ear bone defect. There's also other middle ear and inner ear differences that kids with Alagille syndrome have, and we could study that and learn from that as well. But the more people that participate, the quicker we can actually give you more confident results and get this out in the literature so that more people will be able to learn. And so that's pretty much the slides that I have. B will describe the quick process.
So mine's a little bit less intimidating. So for my testing, what I'm going to do is I'm going to look in your ears, check out your eardrum to see if that's healthy. I'm going to put a little rubber tube or a little rubber piece in your ear, and what that's going to do is put a little puff of air, which you won't even feel, into your ear and wiggle your eardrum. That air is just going to push against your eardrum, and I want to see your eardrum push back. Not going to feel it. You're just going to hear a little hum. After that, then we'll do the actual hearing test. We'll put some headphones on. If you're a tiny little person, we're going to play a game. If you're not a tiny little person, you're going to raise your hand for me when you hear a sound. And basically, the whole point of this is we all hear in here. We know that. But even a mild hearing loss can, and I'm seeing faces that I've tested before. I've tested you. Even with a mild hearing loss, there can still be a huge educational impact. So with a mild hearing loss, you can be not hearing up to a third of the speech sounds, which doesn't seem like a ton, but then you put a kid in a noisy classroom with chairs scooching and teachers moving around the room and teachers talking at the board like this so you can't see their lips move. You're missing about half the speech sounds. So even a mild loss can have a huge educational impact. I think your lives are already hard enough, and my job is to make your life easier. So the more information we have about hearing and hearing loss related to Alagille, the better we can serve you to help increase your quality of life a little bit. So if there's anybody that you think is going to be a little skittish to this, I would love to do my testing before any other testing because I need a lot of patient participation. So if you think someone's going to be a little scared to do some things, hit me up first. So I have a really, really ready to go kind of kiddo. Right.
The other part is that we'll be here all day today and tomorrow. There's sign-up sheets outside with times. You know, if people are busy, you could just give me your number and we could call you when there's a break, or we'll do it late or early in the morning tomorrow. We'll be here all day today and tomorrow. Oh, and if your kid's really young, like last time we tried to test really young children, they may not be cooperative, so we're trying to get kids that are 3 years and older. And if you had your testing done before, we're happy to retest it again. And if you don't have Alagille syndrome and you want your hearing tested, we could also do that as well, just, you know.
Here to serve, people.
You know, I just mentioned at 4:30 I'll be giving a much more detailed talk, including the middle ear loss, but just in general about facial development. We have new zebrafish and mouse models of some aspects of Alagille syndrome, so for those who are interested, at 4:30 in one of the breakout sessions.
And if you've had it tested outside, we'd love to get those results. If you had a CT scan outside, we'd love to look at those as well. The more scans, the better it's going to be for us.
Maybe just a follow-up on that is, at USC where we're at, we actually have an expert, John Go, who is an expert in the temporal bone and specifically in the middle ear bones. And so he's a really good resource to, because these middle ear bones are the tiniest bones in your body, so it can actually be very difficult to really, you have to have an expert who can really identify the shapes in these scans.
Yeah, on that note, you know, one scan was sent to us that was read as normal. I took it to a neuroradiologist, and they said, you know, I think I see some things, but when I showed it to John, he only focuses on the temporal bone, and he clearly defined all the anatomy, which was very helpful. So, you know, just if you're interested, we'd really appreciate your help. Thank you.
All right. Well, in the interest of time, I think we're going to head right into David's talk about itching, cholestasis, and xanthomas, some of our favorite things. And I also, while David's coming up and getting set up, wanted to mention that we have Lumina to thank for all the travel scholarships that I know several families where it helped you come, and that was Lumina sponsoring all of those travel scholarships. They also, well, a couple of the companies that Lumina works with, Premier Research and CRL, are responsible for sponsoring our webcasting and for the little jump drives that you all will be getting tomorrow with all of the presentations on them, so that you can take those home and review those at your leisure. And I'm sure come up with a whole bunch more questions. So I'm going to pass it over to David, and Kira is going to join him to talk a little bit about the itching aspects.
While we're getting set up with the other mic, we will be conducting some interviews here today. No needles, no biopsies, no things in your ears. So I think we're probably the lowest barrier for entry. But these are more opportunities for you and your family to tell your story and your journey with Alagille syndrome. So it's not necessarily similar to the research that we've done before, where we're asking very specific questions about itching or ways you describe itching. It's more if you would like your story to be captured on video and to help others who are not as familiar with the disease understand what it is and what it means to you and how it impacts your life, then we would love to interview you on a nice, comfortable, then either talk to me or Oscar, Pam, or Amy, the rest of the team that are here from Shire. I know I keep saying Lumina. We'll get used to this Lumina Shire transition, but we'd welcome a chat.
Great, thank you. All right, well, it's a pleasure to be back again, and what I'm going to try to do in just a few moments is explain all of the sort of mysteries of bile and bile flow and cholestasis and cholesterol, xanthomas, pruritus, and it's really a tremendously confusing area, and I think even at the level of pediatric gastroenterologists and people who are not really focused in liver disease, there are a lot of complex interactions here, and I'm going to try to summarize them. And towards the end of this, Dr. Kennedy and I are going to team up a little bit to talk about some of the challenges. What you saw in the last hour is sort of the new era of how studies are done and how treatments are developed, proven, checked for safety. I would say to you up front in this talk, everything that we currently use to treat pruritus in Alagille syndrome has been poorly studied or not studied at all, or not studied in Alagille patients. And it's a totally different era when you talk about some of the meds that we're going to talk about that we know work, that are developed for other things. If you go back and you say, show me the studies that prove that this works, you're sort of amazed to find out that somebody reported 6 patients who felt they got better back 15 years ago, as opposed to the science of what is done now. And I think this is both a tribute and a challenge. It's a tribute to better science and the FDA and the NIH and how things really should be done, but it poses tremendous challenges. And for rare diseases, if you didn't have the ability to bring together literally hundreds of patients willing to be studied for long periods of time, nothing would happen going forward. So this is a tribute to Lumina Shire, to children, to the NIH, all of these things coming together. This is the new era, but everything I'm going to show you is based on limited, if any, data actually in Alagille syndrome, although if I had people raise their hands, probably most of you are on most of these things. Would like to say one other thing. We couldn't really say it last night, but everyone in this room, and you're from all across the country, everyone in this room has a representative at the national level, 2 senators, almost everyone in this room has a representative and 2 senators. You know what really, the NIH budget going down is a tragedy for medical science. And so what we really need is for people to write, tell their story, to advocate. It is amazing how one rep or one senator can sort of advocate for this, take it on, and then put something into a bill. And you know, in our Congress, they can put something in an auto manufacturing bill about cholestatic liver disease. I mean, it's really, but that kind of thing then becomes a mandate for the NIH in an advocacy way. And really, if families and relatives of families can't advocate for their own diseases, it's not really clear who will, because in that budget that Dr. Shurker showed you last night, you are talking about everything with AIDS, everything with hepatitis C, everything with, cancer. And so when you get down to diseases that only occur in a couple of people, in small numbers, rare diseases, if the voice is not loud and clear at your own local levels, you just don't get not only the bang for the buck, you just don't get the buck. So the NIH people cannot say that, but they would love it if the country would take more of its huge resources that we spend every year, including on healthcare, and put them into science. But at least as far as what we're going to talk about today, having CLIC, having children, having hundreds of families band together for this kind of research is really the wave of the future. And Dr. Shurker said it just barely last night, but we've just been refunded for a whole nother cycle. And I think we were all concerned that this might not happen. We've had the sequester, we've had the budgetary things. The fact that we still will have a dozen centers working together around Alagille. A bunch of other diseases have been added in. There are an awful lot of liver diseases that aren't in this, that are upset that they're not on the ship, and the ship has left the dock. But it's a really good thing. So with that, I've used up almost all my time. In the rest of the time, we're going to talk about all these different things. So the good news, we did some of these slides last night, and some of you know these. What does the liver do? Whole bunch of stuff. But what we're going to really focus on today, and Eric has given me a laser pointer today, so it's much easier. What we're going to focus on is excreting toxic waste products. Now, we say toxic waste products, but there is one sort of golden nugget in that stream, and that's a biosalt. And a biosalt is a beautiful, spectacular, marvelous, unbelievable compound unless you have too much of them, and unless you have too much of them in the wrong place. And we'll talk about that. So paucity, we know, is a lack of the plumbing, and that plumbing is the conduit. That's what carries everything that the liver puts out into the bile, into the bowel, into the stool, all has to go through this. And patients with Alagille syndrome have sometimes diminished, sometimes markedly diminished number of pipes. The factories, at least in the beginning of life, are still quite good. So we talked also last night about the natural history of liver disease, but just to review, we focus on, is the liver able to support life? Is the liver able to hold blood flow correctly so that it doesn't build up in the spleen and cause bleeding in the esophagus? But far and away, the most common thing, the first thing, and the omnipresent thing for most families, day in, day out, forever, people are not thinking about their albumin. They're thinking about xanthomas and pruritus. And that's overwhelmingly, in the first 10 years of life, the thing that families face the most. So how is Alagille syndrome treated? We talked about some of these other things, and they're certainly all important, but what we're going to talk about today is how to treat the itching and the buildup of compounds that should be excreted, the buildup that you have in the liver and in the rest of the body. So cholestasis, we said, chole, bile, stasis, not going, is a lack of bile flow from the liver. And as we go through this talk, you really have to remember what's in bile. So bile has bile acids, and again, usually really good stuff. And it's actually by actively secreting bile acids or bile salts into this pipe, that's what draws all the water. So if you can't do that well, you have cholestasis, and there's actually a genetic disease where you lack that pump to push things through, and that's another disease that's actually studied by children, by the Childhood Liver Disease Research and Education Network. But so bile acids bilirubin, which is a breakdown of blood. So all of our blood cells last about 120 days, unless you're like a kickboxer or something, and you get bruised all the time. And then when they break down, they give back their iron, and this breaks down to bilirubin, and you have to get rid of bilirubin. You can get rid of a little bit of bilirubin in your urine. So sometimes your children who have very high bilirubin levels have strikingly dark urine and sometimes light stools. But all of the bilirubin in the body is supposed to be processed by the liver, conjugated to something that lets the liver grab it and send it out into bile. So that's a major, major pathway for us. Some cholesterol comes out in the bile. That's our excretion pathway for cholesterol. And then a whole bunch of drugs, toxins, metals, and other things that can be very complicated. The liver really is the area, if you think about it, all the blood flow from the intestines goes to the liver before it goes to the brain, before it goes to the heart, anything else. And the liver looks at all of these compounds and says, I don't know what that is, or that's really bad, or I need to metabolize this. And it has a whole bunch of mechanisms for destroying things coming up from the gut into the bloodstream and then sending them out once it's kind of detoxified them. So these are all issues. We said last night, the liver really is like a very smart, exciting, sexy kidney. So, it's an important place for controlling things. Okay, so cholestasis, what do we measure when we talk about cholestasis? And we do two things. We have these laboratory studies, and as you might imagine, the lab studies are looking at the things that can't get excreted in bile. So we're looking at bilirubin, sometimes bile salts. There are probably many people who haven't had bile salt levels drawn in the last year or two, and the reason in some ways is that it's more important to measure the vitamin levels, which take a couple cc's of blood, rather than the bile salts, which let you know how you're doing, but you're probably already trying to do the best that you can anyway with the bile salts. But we measure bile salts once in a while. There are some enzymes that are pipe duct enzymes, alkaline phosphatase and the GGT or GGTP. We measure those. Those don't really cost us any extra blood. And then from time to time we get cholesterol and all the different types of cholesterol and plot those out. So those are the cholestatic labs. They're different than what we said last night, or the synthetic labs, or the metabolic regulatory labs, or the cell damage labs. We get all these different things, but these are the ones that are like pipe function, what's going on at the pipe level and excretion. And then clinically, what do we see? We see jaundice, we see xanthomas, we have pruritus, itching as a clinical symptom, and then these are also really involved in things like bone disease and poor growth and fat malabsorption, vitamin malabsorption. These are all also related, and while we might be taking care of those directly with other therapies, they're all related to the liver's inability to excrete some of these things, most of which are bad, one of which is really, really good. So cholestasis, and again, when you see a lot of data in these presentations from series that were done in the 70s, 80s, and 90s, when all we ever included were the most seriously affected children. So in these studies, it says that some cholestasis is present in 90% of Alagille patients. It is important to know that you can have cholestasis without jaundice. You can have itching without elevated bilirubin. These are kind of parallel pathways. But, and even last night, up at the podium, people were talking about their cholesterol level versus their itching, and they're not directly correlated, and sometimes surprisingly not correlated. And then these laboratory abnormalities that we talked about a moment ago. So clinically, I'm going to focus first on xanthomas and cholesterol. So this is sort of an important and a little bit complicated slide. But the liver makes cholesterol. So for a long time, we all focused only on not eating eggs, not having, everybody, not just children with Alagille syndrome. But it turns out that the liver makes cholesterol by a very complicated pathway, and there are diseases where you can't make cholesterol, and those are terrible and fatal diseases. The liver then does something with the cholesterol. Cholesterol turns out to be a very important building block for a whole bunch of things. And the liver can secrete cholesterol into blood, where the body uses it for good things, and sometimes sticks it in arteries, which is bad. The liver processes cholesterol from food. So when we eat cholesterol, that goes up through this blood system that 100% goes right into the liver, and the liver deals with lipids on a first pass. The liver excretes cholesterol. It gets rid of some of it, and so that's an important pathway for regulation. And then very importantly for what we're talking about, cholesterol is actually related to bile salts. Cholesterol is the building block for our normal human bile salts. And so that's a very important pathway and one of the important things that the liver does. And there actually are a whole host of diseases in that synthetic pathway from cholesterol making bile salts, where if you have a defect in the enzymes in that pathway, you can't make normal human bile salts, and you can get a progressive liver disease and die. And those are also diseases that are being studied in the children network. They're called bile acid synthetic disorders. Then the liver takes this bile salt nucleus and it conjugates it to something so that most bile salts as they come out are kind of like two big pieces in a way. One piece is sort of fatty or lipophilic, and one piece is water-loving or hydrophilic. And this is what makes these bile salts a detergent. So if you know about soaps or chemistry, the way we dissolve things like oils and stains in our clothes is we treat them with a soap which has a fatty part and a water part, and it dissolves fat molecules. Well, bile salts are essential for taking big fat droplets that we eat and basically breaking them down into little micelles, little droplets that we can absorb. So they're the detergents that are essential for us to absorb fats and fat-soluble vitamins. So the liver does that step, and there are diseases of you can make a bile salt, but you can't conjugate it. So then the liver excretes those bile salts, and they're in the duodenum, in the small intestine, where they help us digest. These things are so hard to make that evolutionarily wise, we decided that we would try to reabsorb them and recycle them. Okay, and that sounds like a great thing, and it is, because they're complicated and we have a way of claiming them back. They go right back to the liver. Sometimes they get reprocessed, and then they get used again. They're only needed for absorption in that 20 feet of intestine. So as soon as the absorptive part of the intestine is done, you try to pull them back. You don't want to pull the bilirubin back because that's just a toxin at this point. But this terminal ileum then resorbs these and brings these back. So understanding that cycle is important for many of the different therapies that we have available, both for high cholesterol and xanthomas and high bile salts and itching, because they are all related in and around this pathway. So you've seen pictures of xanthomas, and sometimes the xanthomas really can be tremendously disfiguring, sometimes painful, and sometimes very prominent, and I think many people in the room have experience with those. I show this just as an interesting progression over time. We said there are some people who have a period of worst cholestasis, and then the xanthomas resolve with time. And so this is in a young woman who was not treated with diversion or transplant or ileal exclusion, but now years later has had all of the xanthomas resolve and is essentially off medication. We can't predict who will take this course and who will take another course, but there are many children who need a bridge to get to the next step. And then this, we're all hoping that the transporter inhibition will be another mechanism that will help us with this, and it may be a bridge to getting us to the next phase of plumbing efficiency, because many people are more efficient at excreting as young adults than they are as infants and toddlers. And obviously a step like a diversion, and certainly a step like a transplant, is either not very reversible, or a transplant, of course, you can never go back and get your old liver if you don't like your new one. So the question is, how will we use these, and how will these things work together? But I think this is just a striking example of going from thousands of xanthomas down to none as time goes on. So we went through a little bit of this. The xanthomas are cholesterol and lipid deposits in the skin. We were talking at breakfast this morning. We really wanted at one point to collect xanthomas, which is a little bit different than what Dr. Lin was talking about. And our IRB, our institutional review board, no matter what we did, would not approve this study because they felt it was too invasive. We just wanted to collect xanthomas, but obviously collecting them means collecting them and analyzing them. For most infants and children, you don't get xanthomas until your total cholesterol starts getting up around 500. And for people where the cholesterol is coming down, they seem to soften and then sometimes go away when you come back around or below that number, if you're lucky enough to come back down on the other side of this. We do tell patients, families, when an infant is 6 months old, that whatever cholestasis they have at that point, it is actually likely to get much worse before it gets better. And I'm sure many of you have had that experience where you see the xanthomas coming over the first 1, 2, 3 years of life. And it's just a normal course of what we see with bile duct paucity. There are different numbers in different studies about what percent of children get xanthomas, and that really depends on how severe their liver disease is. And so what we'll talk about are the strategies that we use to try to eliminate these and the clinical side effect that's associated with them of the intense itching. So one of the things that we use, and again, not really well studied in any pediatric liver disease, but widely accepted, is ursodeoxycholic acid. And I wonder if anybody in here sort of knows where we got this medicine from. Bears. So it actually, in traditional Chinese medicine for a very long time, it was recognized that feeding bear bile or bear gallbladders to people with liver disease could make some of them much better. Who discovered that? But it was right. And so humans make something called chenodeoxycholic acid or cholic acid, but bears make ursodeoxycholic acid. And it's a great bile acid for bears. And bears eat a tremendous amount of fat at one phase of the year, and then they pretty much don't eat much at all for another long phase. Our poor gallbladders, if you didn't eat anything for a month or two, we'd have stones everywhere, But for bears, this is a very adaptive bile acid. And in fact, it works very well for a number of things, but it is a water-loving or a hydrophilic bile acid that drives more water. So it's kind of like turning on the faucets when you turn on your garbage disposal to make sure everything is flushing through. It's not as good for digesting fats, but it has a lot of advantages because it increases bile flow. And so for many people, this is tremendously helpful in decreasing itching. And because we know bile flow is the problem, the early problem in Alagille syndrome, pretty much everyone who's symptomatic ends up going through a phase of URSO or sometimes URSO forever. I'm sure there are people in here who have been on URSO for 10 or 15 years straight. Okay. Now, we talked about the fact that there's this recirculation. And so another strategy for stopping this recirculation, if you think about it in some ways, someone with Alagille syndrome might only excrete 10 or 20% of the normal amount of bile acids in a day because they have a problem with the excretion in the pipes. But once it gets into the gut, the intestine then wants to reabsorb all of it back. And the intestinal reabsorption back is not really broken, so you get a little bit out, and then the body takes it all back because the liver and the skin and the brain are not talking to the intestine about what's really best for the whole body. So there are medications, and these medications were mostly developed a long time ago for people with high cholesterol. And as Dr. Kennedy said, if you have medications that don't get absorbed into the body, there's less chance for toxicity and other problems. And these are not without some problems, and they do taste mostly terrible. And the powders are almost, you know, they're not your favorite. They're tricks. Some people will take them. We heard all the different things last night that people might, you know, hide these medicines in. And then, of course, some of them, as you get older, are in capsules. And as soon as you get to being able to take a capsule, it's a different story. But the point of these medications is whatever bile salts come out into the bowel, you want to try to have all of them bound up, a big sponge that binds them and takes them out in the stool so that the body doesn't reabsorb them, to improve that balance of excretion and reabsorption. So they are good medications. They're used in certain ways, but for a lot of little kids who need them the most, they're really hard to administer. Something like a G-tube turns out to be a major advantage when you're using some of these medications. Okay. So we said that the liver makes cholesterol, and for those of us who are adults, there are medications that inhibit this synthesis of cholesterol, and these are medications that many, many adults are on for lowering their cholesterol level, for prevention of heart disease, prevention of heart attacks after you've had one, preventing a second one, and these are the statins, and they're very, very widely prescribed. Our concerns about these in children are that we know that they can have liver toxicities, and they haven't really been studied in this kind of scenario or shown to be safe, but there are times when they are used with those understandings and provisos where they can help to cut cholesterol synthesis and therefore cuts bile acid synthesis. And people have strong and appropriate and different feelings about how safe they are in different situations, and it really has to be very individualized. But as you saw in that pathway of what goes from the beginning of the liver to the end of itching and bile salts, this is one place where it is interrupted in different patient populations and in adults. So now we're going to move to pruritus, itching. And it's not an itis. You'll see many times itis. It's not inflammatory. But it happens in cholestasis for any kind of liver disease where bile backs up into the body. So there are other genetic metabolic diseases other than Alagille syndrome. There are adult diseases where the bile ducts are destroyed. And as Dr. Kennedy mentioned, there are many more patients as adults who have these diseases than what we have in Alagille syndrome. But in Alagille syndrome, the numbers are actually much more profound in a typical way than many of the people who have cholestasis as adults. But we don't really understand well what causes itching. We know it's some combination of chemicals and skin and nerves and neurotransmitters and the brain. And there are a lot of different models that look at different components of this. And everyone is looking for a great medication that would shut off itching of an allergic type, of a liver type, because it really is, it's a profound and sometimes just ever-present symptom. And certainly in Alagille syndrome, I think people would say of the symptoms, it's different than heart disease or stroke or something like that. It's clearly the worst part of Alagille syndrome. So a lot of science going on in that area, but still not a real great concrete understanding. What we do know in liver cholestasis, for instance, and maybe it goes back to the bear and traditional Chinese medicine, if you have a gallstone and it's stuck in your duct, you get pain, then you get jaundiced, and if it stays stuck there, then you start itching like crazy. If you pop that stone, the itching goes away, the jaundice goes away, and relieving cholestasis clearly can make itching go away. So part of the strategies are directly on itch, part of them are on relieving the cholestasis. And we know that there are neurotransmitters involved, and one of the strategies in treating itching is a strategy to look at opioid receptors. We know that there's a lot of complicated chemicals that are in bile. We don't understand all of them and what they do, but we do know that we really feel itch in our brain more than in our skin. And so there's a lot of research going on at this level now. I'm going to let Dr. Kennedy talk a little bit about how we sort of measure itch, how we think about it, and how that relates to doing studies. So, what are the contributing factors for itching? The bile salts, these other compounds. How good is your liver? There clearly are times when liver function is less, itching seems to be more. These other things like the opioids. There are children who perceive itching all the time, even when their numbers have been great, and there are people who are just the opposite. Skin integrity is key. Once you start to rip open the skin, those scabs and that open skin itch more. And if you think about it, for people who don't have this, like poison ivy, if you itch it and itch it and itch it, it's going to get worse and worse and worse. If you can like freeze it and stay away from it, maybe it settles down a little bit. And clearly in infants and toddlers, everybody I'm sure says, don't scratch. But you scratch, it feels good for a second, and then 5 seconds later you just have to do it more and more. And some of that is also at the skin level. And then there are all kinds of things that affect that: humidity, temperature, and other things, how much soap you use, and so on. And then there are other medications that have some effect here. So we have to think about all these things as we're thinking about treating itching. So it depends on the severity. There are combinations of meds that will work for one person and not for another. You'll have people in here who say this was magic, and someone will say it was a bust, and just the opposite. And it's important to know what did or didn't work at age 1 might be very different than what does or doesn't work at age 3 or 4. So you have to be open to the idea that these things, it's an evolving person with different levels of different things, and something may work differently later on. Then you do have to consider the risk-benefit ratio. We all would love a medication with great benefit and no risk. That would be the holy grail. So just in thinking about the mechanisms, improving bile flow, pushing more things out, improving liver metabolism of these things, binding the bile acids so that they don't get back in, blocking opioids so you don't feel the itch the same way, and antihistamines, which block itch in sort of a different way and a different mechanism. So many times your child might be on one from category A and one from category B and one from category C and so on, and it's the blend and the ratio of those. And sometimes it's interesting and frustrating. A little orso is good, too much orso is bad. Some rifampin is good, too much rifampin could be toxic, and so on. So it really is adjusting all of these different combinations. So this is a list of things, and it's just a list for when you get your very cute little flash drive at the end. It'll have them all there so you can sort of look at them. But we talked a little bit about the therapies that work for cholesterol, where there also are things that work not so much for cholesterol, but for itching itself, separate from the cholesterol level. I'm going to say a few words about some of those. But I do want to say it's important the non-medical therapies for itching can be really important. So things like air conditioning and humidification, ointments, and ointments more than lotions sometimes make the skin dramatically better. Long, hot baths with a lot of soap feel great for a couple of minutes, and then you really pay for them for days afterwards. So you should really try to avoid those. Nobody really wants to hear don't use any soap at all. We're not saying that, but you really do not want to, open up the skin in, warm baths with a lot of soap. Cotton gloves, if you can get them to wear them, it's beautiful. Onesies, it's different if you rub something versus if you can get a fingernail into it. So you want to try to keep the skin covered with something that is clothing where the little kids can't dig into it. The older kids are obviously going to, do what they want to do. And make the fingernails your friend. Don't let them be weapons. Just even a teeny little edge of a fingernail can then cut things open. And once the skin is cut, it just itches so much more. Some people feel strongly that light helps a tremendous amount. But again, all of these things are without significant side effect and should not be overlooked because sometimes they're really great. So medical therapy for itching, we don't use this really for cholesterol, but how many people in here have been on or are on rifampin? So it's a tuberculosis drug. It's a good medication. It's used for other unusual antibiotic reasons. But there was a report a long time ago, first in an adult disease that has some similarities to Alagille syndrome, and then in a small pediatric series that it made itching better. And for how many people in the room has rifampin been really good or magic? Anybody? Yeah. And for how many people was it a total bust? Yeah. Okay. I usually say 50-50. It seems like the eyes, it's 60-40 in this room. But we really don't understand why for some children it's just so dramatically good and for others it's not. And one, sometimes you're not really sure, they're still itching, so you say, let's stop the rifampin now. We've been on it for 2 years. We want to come off. And like 2 weeks later, you get the call, oh, that was a mistake. Can we have another prescription? 90-day supply, 6 refills, but a good medication for many in some scenarios. How many people in here have been on naltrexone or something like that? Okay. For those families, was this excellent, magical, or very good? Can you show of hands? Yeah. And for how many in here was it a bust? Yeah. Okay. And that one also is, I say 50-50, but I'm modifying my small, scientifically poor sample size. But so that's right. Sometimes these medications are also extremely helpful. They don't change the cholesterol. They don't change the xanthomas. But it is the feeling of itch, which really is what is driving the children and their parents, and the lack of sleep, and sometimes the lack of school performance, and all these other things. And so we know that these have a role in some patients. There's nothing about pediatric studies. There's nothing about a pediatric prep. You either get a pharmacy that will formulate this for you, although it's kind of hard to make it as a liquid that they feel confident about, or you sit there with a razor blade on a mirror and you cut these pills up into tenths and then suspend 1/10 of a pill into a teaspoon. It's, you know, it has some analogies to other skills. Okay. So we talked about this, but we know that there are chemicals that make itch felt in the brain, and the analogy that I think is helpful for people as adults who have ever had like Demerol or morphine following a surgery, and all of a sudden you are itching like crazy. You got this medication because you just were cut open for some, you know, you had a baby or whatever, but all of a sudden the itch cascade has gone off. We've known about this ever since these meds were first used, that some people get itchy from them. The blocking of this, that's what these medications do. So these medications, we use them medically when somebody comes in with like a heroin overdose and we want to wake them up because overdose is kind of a bad thing. But we're using these medications to block this central mechanism, and you know, I said sometimes they're really good. So now we're going to move to the surgical things. And the surgical things, all right, I'm going to go through these so quickly then, because you heard about some of these. Okay, biliary diversion, ileal exclusion, transplant. I'm not going to talk about transplant. So this is diversion. This is what it looks like. And what it is to take all of the bile flow and to drain it out into an ostomy bag. It works really well in some diseases and really well in Alagille syndrome much of the time, but not always. It's a sort of step in one direction that's not easily reversed, and it's eliminating a lot of these pruritic compounds so that they can't get recycled. This is what it looks like and how the surgeons do it. I think there's someone in here who might show people, if they were interested, afterwards what it kind of looks like in real life. And Dr. Kennedy showed you a study, and this was one of the early studies, just showing how all the numbers get better in many patients. And so for some people, this is phenomenal. There are some people who have had it done and then got it reversed much later and found that they got much worse. There's a second operation, which we don't do as much, but some people like, which is taking that terminal ileum out of circuit and to avoid this area that resorbs. It's a complicated surgery, but it's internal, okay? And then the ultimate surgery, which works for high cholesterol, xanthomas, absorption, itching, bile salt retention, and that's transplant. And part of our goal is to try to stay away from transplant if we can, but to use it when we need to. So, with that, you didn't think I was going to be able to close that up that quickly? But I think if we could have Dr. Kennedy talk just for 2 or 3 minutes about itching, measuring itching, what you have to do a study that gets a drug approved. You don't need that.
So ultimately, FDA or EMA or whichever regulatory agency you're in the region of will decide if you have proven that your drug is safe and it works. safety is actually the easier of these two to distinguish. I mean, if it's not safe, you know there are certain things that happen. Biochemistry, lab values can start to go awry, or some serious events, obviously the worst of all being a major clinical outcome or death. Those things are somewhat easy to quantify. Does it work is a much more tricky question, particularly when it comes to something like itching. So some of the discussions we've had with the FDA have gone along the lines of, well, itching is a feeling, so only a person can tell you about a feeling. So only a patient can tell you about their itching. But I have yet to meet an Alagille's mom who can't tell you exactly how itchy their child is. So the challenge we have is convincing the FDA that what we're measuring in a clinical trial is actually reflective and associated with the symptom of itching. So, you know, this isn't rocket science in terms of where we ended up, but there's a very distinct, deliberate, and protracted process that you have to go through in order to convince the FDA that, yes, you've done the right research in order to assess, to have a parent assess a child's itching. And this is really important in Alagille syndrome because for the most part, the kids, as you've heard, who have the worst itching are the youngest. So can you have a 4-year-old tell you, rate on a scale from 1 to 10 how itchy you are today? No, that's not going to work. They may pick their favorite number, or they may pick their age, or they may just pick any random number. But what you can say, research has shown, our research has shown, and pediatric patient-reported outcome, which is what these instruments are called, has shown that children around 9, that's when they have the development, the emotional development, the cognitive development to take a question, understand it, pick the option that really represents their experience in that time frame. What we have done is proven through the research that we've done with this community that there are, in fact, 5 ways in which a parent can assess a child's, not their itching, because that's a feeling and only a patient can report on a feeling, but their signs and symptoms or behaviors associated with itching. Again, so it's a little bit of semantics, but ultimately this is what we go through with the FDA. And those 5 ways are, your child tells you if they're verbal, you see them scratching, you see damage on their skin or worsening damage on their skin, you have sleep disturbance, and you have emotional disturbance, so mood disturbance, behavioral signs. So those are that's the foundation of the instruments that we have developed. So we have developed actually a set of 3 instruments to measure itching, and we've gone through this whole process with the FDA. There's a separate division of FDA called the SEAL division that basically oversees all of this type of work. So we have a patient report for itch for pediatric patients, so children who are 9 years of age or older. Sometimes a little bit younger can do it as well, but 9's sort of that line in the sand that we have drawn. We have an observer reporter of itching, so parents or caregivers of children with itching, so that allows them to report on that. And then we actually, for our adult indications, we have an adult patient instrument to report on itching. And the major differences between the adult instruments and the children's instruments are the numbers of gradations on the scale. So an adult can rate, you know, how many times has a doctor said to you, rate from 1 to 10 your pain today? Pain is very commonly rated on a scale of 0 to 10. So itching is rated on a similar scale for adults. Children just cannot distinguish between what's the difference between a 4 and 6. I mean, you have fewer bands, if you like, that they can choose from with bigger differences between those categories. So ultimately, this has been 2 years, many hundreds of thousands of dollars of research to get to a point where the FDA has said, yes, for Alagille syndrome, for these patients, you have an instrument that can be used in a clinical trial, and if you show a change on that instrument, that will equate to a clinically meaningful benefit for your patients. So all those interviews that we've done, all the work that we have done to analyze that data and present it to the FDA has basically led to the point where if we show a change on this instrument, then that's a drug that the FDA has said that they would approve. So it's a complicated process, but it's necessary for good clinical development. And the number of patients we need in order to show that change really depends on how good the drug is. If the drug works as well as diversion surgery, you could probably show that change with a handful of patients. Now, we take a lot of risks in drug development. I'm not quite so ambitious to say that we could show it with a handful of patients. So we are in the process of studying across the world about 60 to 70 Alagille syndrome patients, and hopefully that will be enough to show that this drug on this scale for itching can show an improvement for patients.
Okay, so that leaves us about 4 minutes for a break, which should be right out here outside the doors. And then if you will all get back in here at in about 5 minutes, because in quick succession we have vessels, bones, and kidneys that we're going to be talking about before lunch. What we're going to do is we're going to have the vascular presentation. We're moving, we're going to end up moving the renal health discussion to the 2:30 to 3 o'clock slot this afternoon. And then we're going to move genetics to a breakout. So I will go over those again with you as we get right before lunch so that everybody knows what we're going to do. But I want to get Lisa going here with the vascular presentation so that we can maybe catch up a little bit here. All right, here you go.
All right. Now, can you hear me in the back? Yeah? No, that's all right. This is fine. Sorry for the delay there. My name is Lisa Bergerson. I am not a specialist in Alagilles. However, I am an interventional cardiologist, and I specialize in congenital heart disease and treatment of congenital heart disease, as well as vascular obstructions in the pulmonary artery tree, aorta, pretty much throughout the body. I understand you have a lot of familiarity with liver disease, but you may not have had talks on some of the vascular malformations in the past. And I'm here just to introduce you to some of the vascular malformations that occur, that we see with Alagilles. Many of you won't be familiar with these. Some of you will be. So my talk is in honor of Avery Glenn, who I had the honor to take care of in the year of 2012. I'm going to just briefly talk about Alagilles and congenital heart disease with a focus on vascular disorders seen in some patients with Alagilles. Describe to you some of the techniques that we have available to us in cardiac catheterization to treat these abnormalities, go through a case presentation and a couple concluding remarks. Alagilles, as you know, affects 1 in 100,000 children. It's a multi-system disorder, as you all know, involving the liver, heart, eyes, face, skeleton, and other systems. And many patients have heart or vascular defects. And among those patients, vascular anomalies are common. Congenital heart disease is a common congenital problem. It affects nearly 1% of births annually, being approximately 4,000 infants in the United States per year. Many of our patients are living long into adulthood. There's 1 million patients now who are adults with congenital heart disease. However, it's still the leading type of birth defect associated with morbidity and mortality in our young infants. What do we see with Alagille syndrome? There are structural defects such as tetralogy of flow, atrial septal defects, and valve stenosis. However, more commonly, we see branch pulmonary artery stenosis, sometimes associated with tetralogy of flow, as well as peripheral pulmonary artery stenosis, and more rarely, renal artery stenosis, or a very rare condition called mid-aortic syndrome. Can we dim the lights up front? Would that be possible? So you can see the angiograms. So, thank you. This is a child with Alagille syndrome who presented at 1 year of age. You can, maybe if we just turn them off altogether, they may be able to see them. Presented at one year of age with severe peripheral pulmonary artery stenosis. Many vessels were affected. They underwent multiple cath procedures. And we spent so long trying to get these pictures for you, I want you to be able to at least see them. It's getting better. No. All right, can anybody see those now? Okay, so on the right-hand side of the screen, you have the child at 1 year of age, and I'll show you some normal-looking pulmonary arteries. But you may have to take my word for it, but these are very small vessels, many of them measuring only a couple millimeters in diameter. This causes a lot of work on the right side of the heart because it has to pump against. It has to create higher pressures than normal in order to get through these areas of obstructions. On the left hand of the screen, this is the same child at 10 years of age. They've undergone catheterization procedures, balloon angioplasty, many over the first several years of life. Was doing well at 10 years of age with normal RV pressures. We'll come back to pulmonary artery disease, but I'd like to mention some less common vascular anomalies. One is mid-aortic syndrome. Mid-aortic syndrome is a narrowing of the mid-aorta, and I'll show you a picture of that. It can lead to congestive heart failure, severe hypertension, and stroke without treatment. And as I mentioned, it's a very rare syndrome. However, almost 10% of the patients that we see with this very rare syndrome do have Alagille syndrome. This is a picture of what I'm referring to as mid-aortic syndrome. On the left-hand side of the screen, you see that the abdominal aorta narrows to a very small diameter of only a couple of millimeters. And then on the right-hand side of the screen, you can see some 3D reconstructions of MRI pictures with the similar phenomena. Another less common vascular anomaly compared to the pulmonary artery stenosis is renal artery stenosis. When I say stenosis, what stenosis is an abnormal thickening and narrowing of an arterial or venous wall that results in obstruction, and these obstructions cause increased work on the heart, like I've mentioned. Renal artery stenosis is specifically obstruction to the renal arteries. The true prevalence is really unknown in your patient population of patients with Alagille syndrome. It's been estimated anywhere from 25 to 75% of patients with Alagille syndrome may have obstruction to their renal arteries. This is what that looks like. Here, this is the aorta. These are the renal arteries here. And this is a normal takeoff of a renal artery, whereas this, the right-sided renal artery has severe obstruction proximally. I'll talk to you a little bit about cardiac cath for those of you who are not familiar with it. It's really evolved over the past 20 years from a diagnostic procedure to a therapeutic procedure. In some cases, it complements, but in many cases, we've been able to replace some of the surgical procedures in children. The ages we treat range from premature infants to young adults with congenital heart disease or vascular anomalies. And there's a wide spectrum of procedure types that we perform. They include balloon angioplasty, stent placement for vessels, and then valvuloplasties. We're now doing valve replacements, device closures, and coils. What I'll show you are examples of balloon angioplasty and stents used to treat vascular anomalies. This is my group here. I work on a lot of collaborative, multi-center studies. As like with Alagilles, many of the conditions are very rare and require a lot of collaboration. So here, I'll show you some of the tools that we use. This here is a balloon catheter, and that's inflated from outside the body. There are special balloon catheters that have microsurgical blades on them, called cutting balloons. And Avery was specifically referred to us for our experience using that balloon. This is how balloon angioplasty works. Here's your obstructed vessel. A balloon's inflated inside the body, and it stays open in a larger diameter. When it doesn't stay open in a larger diameter, we have stents that can be placed in the vessels to keep it open in a larger diameter. Some of them are covered and some of them are bare metal. So here you have the obstruction, the stent being inflated, and now it's keeping the vessel open. This is what it would be like in a renal vessel, and I'll show you some angiograms where you have the obstruction, you put the stent in, and it improves the obstruction. This is mid-aortic syndrome repaired with a stent in a larger diameter. Okay. I'm going to now talk about Avery's case. She was diagnosed, as some of you may know, at 18 months of age with Alagille syndrome, and was referred to us in 2012. She had severe hypertension, systemic hypertension, and was on multiple medications because of renal artery stenosis. During her cardiac evaluation, she was also found to have very high right ventricular pressures. So she underwent a cardiac catheterization in Virginia, where her pulmonary arteries were found to be severely obstructed. And then she was referred to us for further treatment. At our initial catheterization, we performed balloon angioplasty of the renal and pulmonary arteries. This was a very complicated procedure. She had some of the worst disease we've ever seen. It was complicated by heart block, pulmonary edema, and she required many days in the intensive care unit with mechanical ventilation before she recovered from that procedure. However, we were pleased to find out and follow up that she clinically had improved and was doing better, had come off some of her blood pressure medications. Her second catheterization was 2 months later. Again, balloon angioplasty, a renal stent was placed. She did very well during this procedure. She had no complications and went home the day after. Again, her blood pressure further improved with relief of the obstruction in the renal arteries, and she had much greater activity level with the reduction in her RV pressure. So I think this is a little bit more illustrative of what I wanted to show in the first picture. In terms of this here would be healthy pulmonary blood flow, large vessels going to a whole distribution of the lung. This was Avery's left pulmonary artery angiogram. And you can see in stark contrast the severe obstructions in multiple segments of this lung. But unfortunately, the right side of her lung was even more severely affected, and that's where we targeted our initial therapy. Here is a balloon being inflated, one of the balloons that I showed you at the beginning. And it shows you that after that balloon dilation, that vessel is larger. There was a lot of extra blood flow that went to that segment, which wasn't seeing blood flow before, and that's why she was so sick after the first procedure and needed mechanical ventilation. This is the renal artery. Similar to the diagram that I showed you, this is an angiogram which shows severe obstruction of the proximal renal artery, narrowing down to the size of this catheter of less than 1 millimeter and then increasing in size past the takeoff. A balloon being inflated here, and ultimately a stent, and I'll show you what that looks like. So here is the stent across the area of obstruction, and then once fully inflated, you can see that area of obstruction has improved. So I think that her case is illustrative of some of the tools and techniques that we use to treat renal disease, as well as pulmonary disease. So Avery came back to us again 3 months later. She had repeat balloon angioplasty and stent placement in the left renal artery, which was also affected. However, that procedure was complicated by a tear in the renal artery and a tear in the pulmonary artery at a subsequent procedure. She required surgical intervention and intensive medical care. Unfortunately, this led to multi-organ failure, and she died in the hospital during that stay. So before I make some concluding remarks, I just wanted to say to all of you, all the families and patients who have Alagilles, how much you touch the caregivers who take care of you. I was inspired by the Glens. They immediately mentioned this group and started talking to me about ways that they could help other people. The strength that I saw with them, as I see with many of the patients that I take care of with congenital heart disease, and I'm sometimes not that great with words, but I'm always astonished with the strength that families can find with children with such important and life-threatening disease, and it's just really inspirational. And I don't know where they find the strength or where you find the strength, but it's inspiring. So in conclusion, catheterization is an evolving field. We have tools available to us that are equipped to address vascular problems if they occur. Alagille syndrome and other genetic disorders that affect children, they encourage our ongoing efforts to develop new technologies and new techniques in our interventional cath procedures. And these minimally invasive treatments for cardiac defects, I have seen and I see substantially improve the quality of life of many, many people. That's it. Thank you.
Okay. All right, so while we get this set up here, our next session is going to be on bone formation and health. And then, like I said before Lisa's presentation, what we're doing is we are moving the renal health discussion to 2:30 to 3 this afternoon in place of genetics. And then the genetics presentation is going to be during the second breakout sessions from 4:30 to 5:30 in 3B in place of the teens only discussion. Because we're going to have the teen and adult concern discussion kind of combined and just do that in the first breakout. And hopefully that will catch us up. Okay.
Hello? 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 Do you want me to just use the microphone?
everybody hear me? No? Can I move it closer? Better? No? Not great. Hello? Good? Okay. All right, great. Thank you so much. My name is Tina Bales. I'm the attending physician at Children's Hospital of Philadelphia, and I am thrilled to be here today, and especially thrilled to see old faces and new faces. I can't believe how quickly your children grow up, and it always makes me happy to see the kids. So thank you. I'm going to talk today about bone health and Alagille syndrome. And I always like to start with the patient. For me, that's really what inspires me, motivates me, and I think really is what drives our research efforts. It really always comes back to the patient. And so I'd like to just talk to you for a few minutes about a child that I met. This was really the first child I ever met with Alagille syndrome years ago. He is a 5-year-old little boy who had really almost all of the characteristic features of Alagille syndrome that we read about in the textbooks and talk about in the lecture. And the folks ahead of me have, of course, gone through all of these features. But really what struck me about him was that when I met him, he came to me in a wagon, and his lower leg was casted. And that was not something that I had read about as being associated with Alagille syndrome. And interestingly, when I talked with his mother, she told me that this was a pattern. And if you look at the history here, you can see that at age 3, he had stress fractures of both the right and lower leg bones, and what we call a buckle fracture, where the leg sort of just bends on the right tibia, which is one of the lower leg bones. And then at 4, he had another fracture of the left tibia, and this really healed with a deformity in the bone. And then at age 5, he presented with a recurrent fracture of that left tibia, and that was how I found him. And then really interestingly, a few weeks later, I saw another child, and he looked almost exactly the same way. So this sounds like the first little boy I told you about, but yet it's an entirely different child, and you can see the left lower leg is deformed. So that really got my interest in bones and Alagille syndrome. And I'd like to just sort of talk to you about a few different things. I'm going to start with some very basics about the skeleton, bone growth in childhood, requirements for healthy bones, and how we measure bone health in children. And then I'm going to talk with you about what we know about Alagille syndrome in terms of weak bones and the data that we've begun to accumulate on fractures in patients with Alagille syndrome. I'll also talk to you about possible reasons for this, ongoing research, and then finally, what can we do to prevent these problems in your children? So just some basics. What does the skeleton do? It supports us. It allows us to stand up straight if we choose to do so. We also find that it protects our organs. So thank goodness we have a rib cage to protect our heart, our lungs, and our vital organs. And then finally, it's a very important reservoir for minerals such as calcium and phosphorus. This is a graph that looks complicated, but I think it's really just helpful to look at one area of this curve here, which I'm not sure how to use this. So, there we go. So you can see this is the area that we focus on in children. You can see this massive increase in bone mass as children age. And then by the time they reach the age of a lot of the young children's parents in this room, you're getting what you call peak bone mass at this point. So your bone mass is as good as it's going to get. And then by the time you're 45, like me, you're a goner. You're in the goner phase. And then when you get to Dr. Bacoli, like it's over. So what do we need to do to maximize this process? How do we make this a child as healthy as possible in terms of the skeletal development? So a lot of this comes down to nutrition. These are just some guidelines. We can make these slides available to you so you can get a sense of what your child might need, but your child needs calcium, and those requirements are going to vary with the child's age, and there are daily recommended intakes that we have. Interestingly, children with Alagille syndrome often need much higher amounts of these things to actually achieve the levels that we need them to achieve. So these are great starting points, and certainly kids need these, but most of your kids will need beyond this. How do we get calcium? Our mothers always told us that we have to drink lots of milk. So dairy products. There are, interestingly, lots of fortified foods out there. So if your child hates drinking milk, you can buy orange juice and waffles that are fortified with calcium. Other foods, broccoli, salmon, sardines, a little harder to get in, but look for the orange juice and the waffles. Vitamin D is critical to bone health. It enhances the intestinal absorption of both calcium and phosphorus, and it promotes the release of calcium from the bone into the bloodstream. It is found in very few foods naturally, and interestingly, most of us make vitamin D from exposure to sunlight. So while we definitely encourage you to use sunscreen, a little tiny bit of sun goes a long way in helping your body to have enough vitamin D. So again, here are some of the requirements for vitamin D, and as time has gone on, even since I was a medical student, we know that people actually need more vitamin D than we anticipated years ago. Food sources of vitamin D, again, your mother told you to drink your milk, but if you look at this slide, you can see that fish is actually one of the best sources of vitamin D. All the oily fishes like salmon, cod, sardines are filled with vitamin D, so if you're lucky enough to get your child to eat these, that's fabulous. There are other important vitamins and minerals, and I think these are important for your doctor to monitor. Vitamin K is helpful in collagen formation. Magnesium is critical for bone health, zinc, and then finally vitamin C. So it's not just all about calcium and vitamin D. Exercise is also critical, and fortunately, if children feel good, they do like to do physical activity. Weight-bearing exercise in particular includes any activity in which your feet and legs are carrying your own weight, and I've listed out some examples, but again, if we can keep kids feeling good, usually we don't have to do a lot of pushing to get this accomplished in the toddler years. So how do we measure bone health in children? There are certainly a lot of tests that we can do to measure the levels of these vitamins and minerals that I talked about, and having a physician who understands the importance of this and really measures these levels carefully is critical. Sorry, I'm having a hard time advancing here. Oh, there we go. Okay. So another technique that we use is radiologic techniques. And some people might ask, well, can we just take a simple x-ray and look at an x-ray and see whether a child has bone demineralization? And you can in the extreme forms, but one of the more sensitive measures is something called a DEXA scan. And if any of you have heard of DEXA scans, most of it is in relation to your 85-year-old grandmother who gets her DEXA scan to look for osteoporosis. This is what a DEXA scanner looks like. It's not really a scary process. It's a little bit like an x-ray, and the child can actually just lie on the table and we take some pictures. So there are centers that have DEXA scanners calibrated for children, and it's important to have the DEXA done at one of these centers. The reason for that is that the scanners are calibrated to make separate measurements for the spine and the whole body, and they report scores differently rather than taking an average for all people as we do in adults and we sort of average everybody out. These scanners are calibrated so that you can look at age and also weight and height as factors that you can use to adjust. And this helps us to be much more accurate in our determination of what the bone density is. So what do we know about bone health and fractures in children with Alagille syndrome? So back in the late 90s, there really wasn't much known at all, and really much less than we know today. I think that what we knew at that point was as much as I knew when I met my first patient, and that was that there were case reports of children who had either severe post-fracture deformities or had seemed to experience fractures at perhaps a higher rate than other healthy children, but there was no data to really support this. So sometime in the early 2000s at CHOP, some folks got together and decided that we should really take a look at this and try to measure what the bone density is in our children with Alagille syndrome related to healthy children. So they looked at 31 children with Alagille syndrome and 80 healthy children and did growth measurements, DEXA scans, and then also looked at how those children were processing fat and how much of that fat that they were taking in was actually not being absorbed well. What they found was that Alagille syndrome and healthy children in that group were similar in age and pubertal status. So there were no significant differences between the children at that level. But what they found was that the children were very small for their age, and they had decreased whole body bone area and bone mineral content for their age, even after we corrected for the height on our sort of super duper pediatric DEXA scanner. And we also found that there was an association between poor bone mineralization and fat malabsorption. So that kind of brings me to my question when I started my fellowship was, well, does that decrease in bone mineral content, bone mineral density, actually translate into more fractures in children? So we looked at this phenomenon in our patients, and interestingly, years before I started, one of my colleagues had mailed some surveys out looking at growth in children with Alagille syndrome. So this was not a survey done to look specifically at bone health, but it asked a few questions about bone fractures that we then capitalized on to sort of find a group of children that we could look at. So they had mailed 402 patient family surveys out, and we reviewed those surveys to identify those patients with a positive history of fracture. And then a few of you are in the audience, and you got a phone call from me, and I reviewed all of the records that we could get our hands on and talked with parents over their phone about their recollection of what had happened. So all told, we got 42 surveys returned. So not a great percentage, but it was something to work with. And we felt that it was valid because it wasn't as though we had asked the families about fractures and only the children who had fractures had emailed back. This was really a survey primarily for other purposes. And what the breakout showed was that about half of the kids were female and half male, and the mean age was about 10 years of age. We found that of those 42 surveys, 12 of the patients, so almost 30%, reported bone fractures. Again, an even distribution. It didn't seem to matter if you were a girl or a boy. And the mean age at fracture was quite young. It was 5 years of age, just like those two children I had initially seen. The total number of fractures in just those 12 patients was 27 fractures, and the average fractures per patient was 2, but we actually had some children with as many as 7 fractures, and I actually know in some of the children I've been referred, the most number of fractures I've ever had in a patient was in excess of 20 fractures. So the mechanism of fracture was very, very interesting. And I circled this because what we found was that most of these were atraumatic, which means that the child had no event that seemed to precipitate it. The mother would say he was standing in the kitchen and all of a sudden he fell, or he walked up the stairs and all of a sudden he was on the ground. Or something more subtle, I suddenly noticed that my child was limping. He wasn't complaining, he was just limping, and I wasn't sure what was going on, and after days I finally took him to the doctor, and this is what they found. So even more interestingly, the distribution of those fractures was very different than what we see in the healthy population. So kids are kids. They will go out and break bones. They climb on the jungle gym. They run and fall, often on an outstretched hand. And the distribution of fractures in healthy children is very well characterized. But what we see in our patients, rather than these outstretched hand type fractures, is actually a preponderance of fractures in the lower extremities. So 70% of the fractures were in the legs. Eight of the total fractures, or 30% of them, were in the femur, the top leg bone, and 11 were in the lower leg bones, either the tibia or the fibula. So what can we take away from this? We know that this survey suggests that children with Alagille syndrome are breaking bones without preceding trauma, that they are experiencing fractures at a younger age than healthy children. So what we generally see is more of the 10 to 11 age group. That varies with whether you're a boy or a girl, but in Alagille syndrome, it's at a much younger age, and that they tend to experience more fractures in the leg bones than healthy children. So we were always interested to quantify things, so we went to the literature and asked how many fractures are there in the healthy population, and there are reports of numbers of fractures in done by patient years, so per 10,000 patient years, and we tried to equate our population to those general numbers, and these are gross estimates. I think that this is kind of preliminary data in some ways, but what we found was that the total fracture incidence in Alagille syndrome patients was 400 out of 10,000 patient years versus 133 per 10,000 patient years in the healthy children. But if you look at the second line, it's even more impressive. There were 128 fractures per 10,000 patient years in the Alagille patients versus only 2.5 in the healthy population. So taken together, that's a threefold increase incidence in the Alagille syndrome patient for all-comer fractures, but a 50% or 50-time increase in incidence in the femur fractures. So why do children with Alagille syndrome have weak bones? I think there are a lot of reasons. One of them is probably poor intake. A lot of the children are not hungry. They don't seem to eat as well. There are things that we can do to address that. Some of the families have opted to use feeding tubes to help augment the child's appetite, and this can be extremely effective, not only in translating into improved growth, but also into improved bone health for the children. And I can tell you in my experience, most of those families were very reluctant to do that kind of feeding because it seemed unnatural to them, but yet when they saw the outcomes and how their children's overall development improved, they were very much in favor and recommended it to a lot of the other families. We also know that just malabsorption due to the liver disease, and some children actually have some effects in the pancreas, can cause deficiencies in absorption of calcium and fat-soluble vitamins, including vitamin D and K, which are critical for bone development. Kidney disease can also have its own effects on the bones. So if your child has intrinsic renal disease, that can also be impacting their bone health. And then finally, some children are exposed to different medicines for other reasons. So medicines like steroids, immunosuppressants used after transplant, And then cholestyramine can be a very useful drug for some purposes in terms of binding all those bile acids, but it also tends to bind up the fat-soluble vitamins like vitamin D, and so you need to really account for that and help to make sure that the child's getting adequate vitamins. We also thought perhaps there were other reasons, because we have other children with severe liver disease who don't eat well and have all of those characteristics that I talked to you about on the previous slide, but yet they didn't seem to have the severity of presentation that we see in the Alagille syndrome population. So we postulated that perhaps there's something about bile acids or bilirubin that is almost toxic to the bones. I don't think anybody's really proven this, but it's a thought. Other thought would be, is there something about the change in the gene, the JAG1 mutation, that's actually affecting the developing bone, not just in terms of the anomalies that we see, like little butterfly vertebrae that really often have little to no consequence, but really affecting the intrinsic structure of the bone and perhaps making it even more vulnerable to these changes. So I think that there is evidence suggesting a gene effect. These are two papers that came out several years ago in Nature Medicine, which is really the be-all, end-all journal of at least of medicine, Nature and Nature Medicine. And these showed that Notch signaling, the Notch pathway where the JAG1 gene and the Notch2 gene lie, are actually important in regulating cell development within the bones. And some of my research in the lab, which some other folks have ongoing now, has looked at exploring the effects of that mutation in the developing bones. So we actually created a mouse. This is a skeleton of a tiny baby mouse embryo that's been stained. And you can actually use genetic techniques to just simply delete the JAG1 gene or change that gene in just one cell in the bones. And we did that, and we found that the regulation of the cell components were different in those bones than they were in other healthy mice. So exactly what that means, I'm not really sure, but I think that it suggests that your children may be at higher risk for bone problems than just simply the nutritional aspects lead us to believe. So what does that mean to all of you, and to what does it mean to your kids, and how can we protect them from bone fractures in the future? So I think that one thing is just being aware of the possible signs of fracture, and this seems simple, but a lot of the parents that came to me were horrified that their children had fractures. They never in their wildest imagination dreamed that they would have a fracture, and so they felt bad that they'd waited 2-3 days to take the child to the physician, and I think that's natural. I think that it's important to let families know that this can happen, so that if your child is complaining or limping or refusing to walk, that you know that this could be a problem. The other is to talk to your doctor about getting a DEXA scan on a pediatric scanner. The age of the child is usually about 4 or 5. Before that, they're a little too rambunctious to want to sit still in the scanner, but it's a fairly short procedure, and a 4 or 5-year-old can do this. I also suggest that families talk to their doctor about measuring vitamin and mineral levels. I usually do this every 3 months. As the child's growing very rapidly, it's important to keep up with their needs, and so even though you have a normal level one time, you may find that it's going to take you a lot of work to keep up with the child's growth requirements. I often will back off if I keep finding normal levels after a while, but some kids really constantly have abnormal levels, and you have to really chase those. I also suggest that you talk to your doctor about looking for kidney disease. We've had a lot of kids come to us where the physicians aren't aware of this phenomenon. It can affect the appetite, so if children have low bicarbonate levels, they often aren't hungry, and they may not have obvious signs of kidney disease. These are subtle findings on the labs that, if you're used to looking at this, that can be helpful. Encourage safe weight-bearing exercise. As I said, most people with 3-year-olds don't need to encourage that if their kids feel good. And then incorporating all of the sources of calcium and vitamin D. If your child is not growing well, I suggest getting a dietitian involved, because I think, especially in some of the major pediatric centers, these people who are trained to look at pediatric diets are excellent, and I've partnered with those folks in many of my patients and found tremendous help. Supplement vitamins as needed, and I always mention that your doctor can help you get insurance coverage. I think a lot of families get frustrated because the medicines are expensive and the insurance will cover rifampin, but they won't cover the vitamin E, but yet the vitamin E costs $100 a month. And so if your doctor can write a letter explaining the necessity of this for your child, which we actually have templated letters that I'd be happy to send you if you come and find me after the meeting, we can have you give that to your doctor, and they can write, sign the letter, and that can sometimes be helpful. And then finally, seek care of an experienced orthopedic doctor if your child does have fractures, because I think it's important for them to understand that your child may have some underlying metabolic bone issues that might make that treatment slightly different than it would if the child was a typical healthy child. I'd like to thank my colleagues at Children's Hospital. I also had some wonderful mentors at the University of Connecticut Children's Medical Center who were nice enough to let me come to their lab when I was a fellow and teach me some of their techniques, and I'm forever grateful to them. And also, in particular, the families that not only participated in our studies, but came to our center and really trusted us with their children. I'm very appreciative. Thank you.
Okay, so I think we're at 12:05, which means that we are into lunch. I'm sorry that you guys have not had a chance to ask much in the way of questions, but maybe we can do a little bit more of that this afternoon, depending on how things go. So we have lunch from 12 to 1, and then we're doing a group picture from 1 to 1:30. Anna, do you know where that is going to be? Out on the patio? Okay. So we're going to gather on the patio after lunch. Bring the kids along. We really want to have them in the picture, and enjoy lunch. And we'll see you back in here at 1:30.